This Bioinspired forum, the idea of this Bioinspired forum actually came originally from the Biomime communication team from Mats Broden and Susanne Rosen. And I would like to start thanking them for organizing this, but also, of course, our co-hosts, sponsors, everybody that has contributed to this organization, and of course to all of you for coming here. This is a great day where we have a good combination of different expertise, and I think we should really exploit this for developing biomimicry and biomimetic discussions. When our communication team started to work in Biomime, the first thing they did was to go around all the scientists in Biomime and ask these two questions to each one of them. How can your research be useful in future product development and design? And we work a lot with cell walls and plant cell walls in particular. Why is this interesting with the perspective of developing products? And I'm going to try to address and answer these questions by giving you flashes of the activities and some of the results we have obtained over these 3 last years in the Biomime Center. This center was originally initiated by Tula Terry, the former director of this center, and she gathered this constellation with a node at the Umeå Plant Science Center where a lot of the wood biology is done, which is a key activity, of course, in our center. And then we have experts in a whole range of activities from the departments at KTH, fiber and polymer chemistry, surface chemistry, the Research Institute in Vincent, and also, of course, the School of Biotechnology. What we have as a vision is to exploit nature and plant biomass in particular to move towards new products. So this is our long-term vision. And are we far from there? This is what I'm going to try to address here, and maybe we will come back to this slide at the end to open the discussion. One of the materials we want to use is wood. Wood is traditionally used for energy, construction, pulp and paper. But we see a great potential there for novel products, particularly in the biopolymers and biomaterials area, and this is the focus of the center. The knowledge you need to build these biomaterials is the same fundamental knowledge as you need to break it down, for instance, for the production of biofuels. And some of us in Biomime have projects also running in this area. Wood is a fantastic material because it's a highly hierarchical structure. It's a very complex structure, in fact. And if you take any plants, like those that are standing there, and you take any tissue from this plant, in every single one of the different tissues, the roots, the stem, whatever, you have cells which are delimited by a cell wall. And it is the cell wall that gives the properties of the wood. And what this cell wall is a material composed of rather resilient and strong fibers, which are highlighted in the corner there. And these strong fibers, these strong cylinders, are connected to each other by a soft material, which are also carbohydrates. And these things evolve during plant growth. And we have here a natural composite that maybe can be tuned to obtain a material with better property. And this is what we're trying to do in Biomime. We're trying to understand how this natural composite is formed, how it is remodeled, and exploit this knowledge to develop materials from this system. We will have a presentation later about multidisciplinary research and the challenges related to these aspects. Obviously, Biomime has had to go through these challenges. We have to work at the level of the organism, down to the tissue, the cells, the proteins, and the genes, down to the molecular level. And then we want to exploit this knowledge for materials development through the production of genetically modified organisms, combined with chemistry and material sciences. Obviously, we need a very strong multidisciplinary expertise. And now I think Biomime has been running for 3 years, and I will show you a few examples that illustrate that this is possible and that this is really on the right way. So in the rest of my presentation, I will give you flashes of some of the research and the potential applications. What is drawn here again in the box is again these cylinders, these cellulose cylinders, which are connected by this soft material, these soft carbohydrates. And one of the systems that has been exploited is an enzyme which is naturally occurring in the plant cell wall and which is able to cut these cross-linking soft materials between the cellulosic fibers and join them again together. And this is how the cell wall and the plant cells expand in the plant. How can we use this fundamental knowledge for biotechnological application? Well, we can take the same enzyme, produce it in the laboratory, and mix it with two polymers, one which is called xyloglucan up there, which is a natural polymer, and another one which is shorter, but which we can modify chemically. And then we can use the enzyme to join these two polymers together. It's its natural activity. And then we mix up this activated xyloglucan with a cellulosic matrix. And the advantage of doing that is that you won't touch at all the mechanical properties of cellulose because you don't modify the cellulose itself. But you will be able to coat the surface of the cellulose with these activated groups. What this means concretely is this. Yes, chemistry, but this is very useful. For instance, if you look at this hydrophobic paper up there, what is it? It's just this concept that has been exploited to graft a polymer which is hydrophobic at the surface of a cellulosic matrix. And when we do that, again, we don't lose any of the mechanical property of the cellulosic matrix. If you graft a compound which is fluorescent, you make fluorescent paper. If you graft a substrate of another enzyme, which then can be converted into something that you can see by the naked eye, which is stainable, then you can make a biosensor and detect enzyme in biological media. There are a whole range of applications like that. These are only a few examples. And this is a typical example where multidisciplinarity has been essential in biomime. All this started with fundamental research in the biology, biochemistry area, and you can see that it has been coupled to chemistry, organic chemistry, synthetic chemistry, and surface chemistry to develop these interesting potential materials. The good thing with this system is that it can be applied to any types of cellulose, from soft and hardwood pulps, cotton fibers, regenerated cellulose, even bacterial cellulose. And you can see the whole range of potential applications in a whole range of areas: textile, packaging, etc., even in the biomedical area. Surface chemistry is obviously something very important in biomime. And here are a couple of examples more. By using surface chemistry, we can, for instance, coat the surface of a paper by polymers which like to interact together and that can form these multi-layers. And if you do that, you can get a paper with dramatically increased strength. That's another example of soft surface chemistry. You can also try to engineer the surface to have them responsive to different stimuli. And this is an example. What is represented here is a polymer grafted at the surface of a matrix. And on your left-hand side, the polymer is fully extended because it is stabilized by chemical bonds. These chemical bonds can be broken, for instance, under the effect of temperature and pH. And if you break them, the polymer might collapse. And this is what you see on the right-hand side, top right-hand side. And if the polymer collapses, and if it's hydrophobic, by having changed the pH, you have changed the property of the surface of the material, and you have turned it into something more hydrophobic in response to the stimuli, pH, temperature, for instance. We don't use only plant systems. We also use microbial systems.